Good evening, everyone, and welcome to the Droning Professor. Tonight we're going to learn about how your brain falls asleep. So as you make yourselves comfortable, please take a moment to let us know where you're listening from and feel free to subscribe. Now, as always, take a moment for some deep breaths, letting your mind and body relax for a good night of sleep. Tonight, we embark on a journey into one of the most remarkable phenomena in all of biology: the transition from wakefulness to sleep. This is a journey you have taken approximately 12,000 times in your life so far, give or take. And yet, I would wager that most of you have absolutely no idea what is actually happening inside your skull during those strange, twilight moments when consciousness begins to dissolve into dreams. You have been doing this your entire life with the same casual indifference with which you breathe or blink, completely unaware of the neurological symphony occurring just behind your eyeballs. So, tonight, we pull back the curtain, we examine the machinery, and hopefully by the end of this lesson, you will never look at your pillow quite the same way again. Let us begin with the fundamental question that has puzzled philosophers, scientists, and exhausted parents since the dawn of humanity. Why do we sleep at all? This seems like a reasonable place to start, does it not? After all, sleep is genuinely bizarre when you think about it. For roughly 1/3 of your life, you lie down, close your eyes, become unconscious, and remain completely vulnerable to predators, weather, and whatever Netflix show your roommate decides to watch at maximum volume. From an evolutionary standpoint, this seems like a spectacularly bad idea. And yet, every animal with a nervous system sleeps. Fruit flies sleep, jellyfish sleep, and they do not even have brains, which honestly seems unfair. Even the humble roundworm, with its 302 neurons, takes naps. Sleep is so universal, so deeply conserved across the animal kingdom, that it must serve some absolutely essential function. Or rather, functions plural, because as we shall see, sleep does many things for your brain, and almost all of them are things you would very much like your brain to do. But before we can understand why we sleep, we need to understand how we fall asleep. And this requires us to meet the cast of characters in your brain who are responsible for this nightly miracle. The first thing you need to understand is that sleep is not simply the absence of wakefulness. This is a common misconception, and it is completely wrong. Sleep is not what happens when your brain gets tired and shuts down like a laptop with a low battery. No, sleep is an active process. Your brain does not just stop doing things when you fall asleep. Rather, it starts doing different things. It switches from one mode of operation to another, like changing gears in a car, except the car is made of approximately 86 billion neurons. And the gears are controlled by a cocktail of neurotransmitters that would make a pharmacist weep with confusion. The transition from wakefulness to sleep is orchestrated by two primary systems in your brain, and they work together in a beautiful and complicated dance that scientists have spent decades trying to understand. These systems are called Process S and Process C, which sounds like the names of experimental military aircraft, but I promise they are much more interesting than that. Process C is your circadian rhythm, the internal 24-hour clock that tells your body what time it is. This clock is controlled by a tiny structure called the suprachiasmatic nucleus, which sits in your hypothalamus, right above where your optic nerves cross. The suprachiasmatic nucleus contains approximately 20,000 neurons, which might not sound like many when your brain has 86 billion of them. But these 20,000 neurons punch far above their weight. They are the master timekeepers of your entire body, coordinating everything from when you feel hungry to when you feel sleepy to when your body temperature rises and falls. The suprachiasmatic nucleus, which scientists mercifully abbreviate as the SCN, receives direct input from special cells in your retina called intrinsically photosensitive retinal ganglion cells. These cells contain a photopigment called melanopsin, and they do not care about what you are looking at. They do not help you see shapes or colors or the face of your beloved. Their only job is to detect ambient light levels and report this information directly to the SCN. This is how your brain knows whether it is day or night, even if you have been staring at spreadsheets in a windowless office for 8 hours. When light levels are high, the SCN sends signals that promote wakefulness. When darkness falls, the SCN changes its tune and begins orchestrating the release of a hormone you have probably heard of, melatonin. Melatonin is produced by the pineal gland, that mysterious structure that the philosopher Descartes once believed was the seat of the soul. We now know it is not the seat of the soul, but rather the seat of sleepiness, which is perhaps less romantic but considerably more useful. Melatonin secretion typically begins about 2 hours before your normal bedtime, rising sharply as darkness deepens, reaching its peak around 3 or 4 in the morning, and then declining as dawn approaches. Melatonin does not knock you out like a sedative. Instead, it opens a biological window during which sleep becomes possible. It is less like a sleeping pill and more like an usher in a theater, quietly guiding you toward the door that leads to sleep, but not actually pushing you through it. Now, here is where things get interesting. Process C, your circadian rhythm, does not actually make you feel sleepy. What it does is modulate your alertness throughout the day in a predictable pattern. Most people experience peak alertness in the late morning and early evening, with a dip in the early afternoon, which explains why so many cultures have invented the siesta and why so many corporate meetings scheduled for 2 p.m. are absolute disasters. The actual feeling of sleepiness, that growing pressure to close your eyes and surrender to unconsciousness, comes from process S, the homeostatic sleep drive. And process S has its own special molecule, adenosine. Adenosine is a fascinating little chemical. It is a byproduct of cellular metabolism, produced whenever your neurons burn energy. The more active your brain is during the day, the more adenosine accumulates in certain brain regions, particularly the basal forebrain. As adenosine levels rise, they bind to receptors on neurons that normally promote wakefulness, and they tell those neurons to quiet down. The longer you have been awake, the more adenosine builds up, and the sleepier you feel. This is your sleep pressure, and it increases steadily from the moment you wake up until you finally go to bed. Now, I know what some of you are thinking. You are thinking about coffee. You are thinking about that beautiful, aromatic, life-giving beverage that has gotten you through more mornings than you care to count. Here is the secret of coffee. Caffeine is an adenosine receptor antagonist. This means that caffeine molecules have a shape similar enough to adenosine that they can bind to the same receptors, but when they do not activate those receptors. They just sit there, like a key that fits into a lock but cannot turn. As long as caffeine is occupying those receptors, adenosine cannot get in, and you do not feel sleepy. But here is the catch, and it is an important one. Caffeine does not reduce your adenosine levels. It does not eliminate your sleep pressure. It merely hides it from you. While you are feeling alert and productive thanks to your third espresso, adenosine is still accumulating in your brain, building and building behind a chemical dam. And when the caffeine finally wears off, which it does with a half-life of about 5 to 6 hours, all that adenosine comes flooding back. This is the infamous caffeine crash, and it is worse than if you had never had caffeine at all, because now you have even more adenosine waiting to make you sleepy. I am not telling you not to drink coffee. I am a reasonable person. But I am telling you that if you want to understand how sleep works, you need to understand that caffeine is not giving you energy. It is borrowing energy from your future self, and eventually that debt comes due. So we have process C, your circadian rhythm, which determines when your body thinks sleep is appropriate, and process S, your homeostatic sleep drive, which determines how much sleep pressure has accumulated. When these two processes align, when your circadian rhythm says it is nighttime, and your adenosine levels say you have been awake long enough, the conditions are right for sleep to occur. But conditions being right is not the same as actually falling asleep. For that, we need to look at the specific brain structures that flip the switch from wake to sleep. Deep in your hypothalamus, there is a tiny cluster of neurons called the ventrolateral preoptic nucleus, or VLPO. I want you to appreciate how small this structure is. In a human brain, it contains perhaps a few thousand neurons. Your brain has 86 billion. The VLPO is like a single light switch in a building the size of a small city. And yet, this light switch controls whether the city is awake or asleep. The neurons in the VLPO are sleep-promoting neurons. They contain two inhibitory neurotransmitters, gamma-aminobutyric acid, which everyone calls GABA because life is short, and no one wants to say gamma-aminobutyric acid more than once, and a neuropeptide called galanin. When these VLPO neurons become active, they send inhibitory signals to all the brain regions that promote wakefulness. They tell the wake-promoting systems to shut up and sit down. And there are many wake-promoting systems. The brainstem contains clusters of neurons that release norepinephrine, serotonin, and dopamine. The hypothalamus contains neurons that release histamine, which is why antihistamines make you drowsy. The basal forebrain releases acetylcholine. And a particularly important group of neurons in the lateral hypothalamus releases orexin, also called hypocretin, which is one of the most powerful wake-promoting chemicals in the brain. During wakefulness, all these systems are active, and they collectively suppress the VLPO. But as night falls and adenosine accumulates, the balance shifts. The wake-promoting systems begin to lose their grip, and the VLPO neurons start to fire. Once they begin firing, they inhibit the wake-promoting systems, which means those systems can no longer inhibit the VLPO, which means the VLPO fires even more strongly, which inhibits the wake-promoting systems even further. This creates what scientists call a flip-flop circuit. Just like a light switch that is either on or off, never in between, this circuit tends to push the brain rapidly from one state to the other. You are either awake or asleep, with minimal time spent in the transition. This is actually very useful, because spending a lot of time half awake and half asleep would be dangerous for any animal. You want clear states, alert and ready for action, or fully unconscious and recovering. The flip-flop circuit ensures that transitions happen quickly and cleanly. Of course, those of us who have ever spent 45 minutes lying in bed unable to fall asleep might dispute the word quickly, but I assure you, from a neurological perspective, it really is quite fast. Now, let us talk about what actually happens in your brain as you transition from wakefulness to sleep. This is where things get genuinely strange, and I find it absolutely delightful. The first thing that happens is that your core body temperature begins to drop. This starts about 2 hours before sleep onset, orchestrated by your circadian rhythm. Your blood vessels dilate, particularly in your hands and feet, allowing heat to radiate away from your body. This is why your extremities often feel warm before bed, even as your core temperature is falling. It is also why a warm bath before bed can help you sleep, counterintuitive as that might seem. The warm water causes your blood vessels to dilate, and when you get out of the bath, you lose heat rapidly, accelerating the natural temperature decline that triggers sleep. The drop in core temperature is not just a side effect of sleep. It is actually part of the mechanism. The VLPO neurons that promote sleep are also involved in thermoregulation. When they activate, they do not just tell the wake centers to be quiet. They also initiate the physiological changes that lower body temperature. This is why fever can disrupt sleep so profoundly, and why sleeping in a cool room, around 60 to 67 degrees Fahrenheit, is generally conducive to good sleep. As your temperature falls and the VLPO gains control, your brain waves begin to change. During alert wakefulness, your brain produces fast, irregular electrical activity called beta waves, oscillating at frequencies of about 13 to 30 cycles per second, or hertz, if you prefer the scientific term. As you relax and close your eyes, these give way to alpha waves, slower and more regular, at about 8 to 12 hertz. You can think of this as your brain beginning to idle, like a car engine at a red light. Then comes the threshold of sleep. As you begin to drift off, you enter what sleep scientists call stage N1, or light sleep. This is the hypnagogic state, that strange borderland between waking and dreaming. Your brain waves slow further, becoming theta waves, at about 4 to 7 hertz. Your muscles begin to relax. Your eyes may roll slowly under your closed lids. This is also when you might experience some of the weirdest phenomena associated with sleep. Some people see geometric patterns or fleeting images, fragments of dreams that have not yet fully formed. Some people hear sounds that are not there, snippets of music or voices. And many people, perhaps 60 to 70% of the population, at some point in their lives, experience hypnic jerks. A hypnic jerk is that sudden, startling sensation of falling or tripping, accompanied by a full-body twitch, that yanks you back to wakefulness just as you were about to fall asleep. These are harmless but extremely annoying, especially if you have been trying to fall asleep for a while. Scientists are not entirely certain why they happen, but the leading theory is rather charming. As your muscles relax during the transition to sleep, your brainstem may misinterpret this relaxation as falling. After all, from the perspective of your ancient primate brain, sudden muscle relaxation while you are upright usually means you are about to plummet from a tree and die. So the brainstem, in a moment of evolutionary overzealousness, fires a signal to all your muscles at once, a kind of emergency wake-up call. You jerk, your heart pounds, and you have to start the whole falling asleep process over again. If it makes you feel any better, hypnic jerks are more common when you are overtired, stressed, or have consumed too much caffeine. So, if you are experiencing them frequently, your body is probably trying to tell you something. Assuming you successfully navigate the hypnagogic waters without being thrown back to wakefulness by your paranoid brainstem, you will progress to stage N2, which is considered true sleep. Stage N2 accounts for about 45 to 50% of a typical night's sleep in adults, making it the most common sleep stage. Your brain waves continue to slow, but now something new appears. Sleep spindles and K-complexes. Sleep spindles are brief bursts of rapid brain activity, lasting about half a second to 2 seconds, oscillating at about 12 to 14 hertz. On an electroencephalogram, they look like little spindles of thread, hence the name. They are generated by a circuit involving the thalamus, that central relay station of the brain, and specifically a structure called the thalamic reticular nucleus. Sleep spindles serve multiple functions. They appear to help maintain sleep by blocking external stimuli from reaching the cortex. They also seem to be involved in memory consolidation, helping to transfer information from short-term to long-term storage. Interestingly, people with higher sleep spindle activity tend to score better on certain tests of memory and learning, and people who take naps that include spindle-rich sleep show improved performance on tasks they learned before the nap. K-complexes are even more dramatic. They are the largest electrical events in the healthy human brain, big, slow waves that stand out on an EEG like mountains on a map. K-complexes are generated in the cortex, particularly the frontal regions, and they seem to serve a protective function. When an external stimulus occurs during sleep, a sound or a touch, the brain often generates a K-complex in response. This appears to be the brain's way of saying, "I noticed that, but it is not important enough to wake up for." K-complexes suppress the arousal that would otherwise follow the stimulus, helping you stay asleep despite the noise of your snoring partner or the ambient sounds of your environment. After stage N2 comes the deepest stage of sleep, called stage N3, or slow-wave sleep. This is when your brain waves become their slowest and largest, delta waves, oscillating at less than 4 hertz. During slow-wave sleep, your brain activity becomes highly synchronized. Millions of neurons fire together in great, rolling waves of activity, then fall silent together, then fire again. On an EEG, this produces the characteristic slow, high-amplitude patterns that give this stage its name. Slow-wave sleep is the most physically restorative stage of sleep. This is when your body releases the most growth hormone, when your immune system does its most intensive repair work, when your muscles and tissues recover from the day's exertions. It is also, remarkably, when your brain takes out its garbage. This brings us to one of the most exciting discoveries in sleep science in recent years, the glymphatic system. For a long time, scientists were puzzled by the fact that the brain, despite being one of the most metabolically active organs in the body, appeared to lack a lymphatic system. The lymphatic system, in the rest of your body, is responsible for clearing waste products from your tissues. How was the brain managing without one? The answer, discovered in 2012 by a team led by Macon Niedergaard, is that the brain has its own unique waste clearance system, which they named the glymphatic system, with a G, because it depends on glial cells, the brain's support cells. During wakefulness, this system operates at a relatively low level. But during sleep, particularly during slow-wave sleep, something remarkable happens. The spaces between your neurons expand by as much as 60%. Cerebrospinal fluid floods into these expanded spaces, flowing along pathways that run alongside your blood vessels. As it flows, it picks up waste products, including a protein called beta amyloid, which is one of the hallmarks of Alzheimer's disease. The cerebrospinal fluid then drains out of the brain, carrying all this neural garbage with it. In 2024, researchers confirmed this system exists in humans, not just in the mice where it was first discovered. They also discovered that the glymphatic system is driven by specific patterns of neural activity during sleep. When neurons that fire together during slow-wave sleep synchronize their activity, they create waves of blood flow that pump cerebrospinal fluid through the brain. Scientists have summarized this with a phrase I find rather poetic: neurons that fire together, shower together. The implications of this discovery are profound. It suggests that one of the fundamental functions of sleep is to clear away the metabolic waste that accumulates during waking brain activity. It also suggests that chronic sleep deprivation could contribute to the accumulation of toxic proteins in the brain, potentially increasing the risk of neurodegenerative diseases. Indeed, studies have shown that even a single night of sleep deprivation leads to increased levels of beta amyloid in certain brain regions. The trash collectors have not come, and the garbage is piling up. But the brain does not stay in slow-wave sleep all night. After spending perhaps 30 to 40 minutes in the depths of stage N3 during your first sleep cycle, you begin to ascend through the lighter stages. Stage N2 again, perhaps a brief return to N1, and then something entirely different happens. You enter rapid eye movement sleep, or REM sleep. REM sleep is strange. It is so strange that when it was first discovered in 1953 by Eugene Aserinsky and Nathaniel Kleitman, they initially thought their equipment was malfunctioning. During REM sleep, your brain waves look almost like they do when you are awake, fast and irregular. Your brain is extremely active, consuming nearly as much oxygen as during waking. Yet you are deeply unconscious, in some ways more difficult to wake than during slow-wave sleep. And then there is the paralysis. During REM sleep, your voluntary muscles are almost completely paralyzed. This is called REM atonia, and it is caused by specific neurons in the brainstem that send inhibitory signals to your motor neurons. Your diaphragm keeps working, so you can breathe, and your eye muscles remain active, darting back and forth in the rapid eye movements that give this stage its name. But the rest of your body is essentially frozen. This paralysis is a safety mechanism, and it is extremely necessary, because REM sleep is when you dream, and dreams, as you may have noticed, often involve physical activity. Running, fighting, flying, doing whatever it is that your unconscious mind comes up with. If you were not paralyzed during REM sleep, you would act out your dreams, and this would be both dangerous and extremely inconvenient for anyone sleeping near you. Some people do lose this paralysis due to damage to the brainstem regions that control it. This condition is called REM sleep behavior disorder, and people with it do exactly what I just described. They punch, kick, jump out of bed, and occasionally injure themselves or their partners. In experiments with cats, researchers found that lesioning the specific neurons responsible for rematonia produced animals that would stalk, pounce on, and fight with imaginary prey during REM sleep. While their brain activity clearly showed they were still asleep, they were acting out their dreams, and it is both fascinating and slightly terrifying to watch. The chemical responsible for much of what makes REM sleep distinctive is acetylcholine. Remember those basal forebrain neurons that release acetylcholine to promote wakefulness? During REM sleep, similar neurons become highly active again. Acetylcholine floods the brain, creating the activated brain state that makes vivid dreaming possible. Meanwhile, the neurons that release norepinephrine and serotonin, which are active during waking, go almost completely silent. This unusual neurochemical cocktail, high acetylcholine, almost no norepinephrine or serotonin, may explain some of the peculiar qualities of dreams, their emotional intensity, their illogical quality, their remarkable forgettability. The absence of norepinephrine during REM sleep is particularly interesting. Norepinephrine is involved in memory encoding, in laying down new memories. Its absence during REM might explain why we forget most of our dreams so quickly after waking. The machinery for making new memories is simply not running. REM sleep appears to be important for emotional processing and for certain types of memory consolidation, particularly procedural memory, the kind of memory involved in learning skills. People who are deprived of REM sleep show impairments in creative problem-solving and emotional regulation. Their brains do not seem to properly process the emotional experiences of the previous day, leading to increased anxiety and irritability. And dreams. What are dreams? This remains one of the great mysteries of neuroscience. We know dreams occur predominantly during REM sleep, though they can occur during other stages as well. We know dreams involve activity in the visual cortex, the motor cortex, the emotional centers of the limbic system, and the memory regions of the hippocampus. We know that the prefrontal cortex, the part of your brain responsible for rational thought and self-awareness, is relatively quiet during dreams, which may explain why dreams so rarely seem strange while we are having them. We accept the most absurd scenarios as completely normal because the part of our brain that would normally notice the absurdity has clocked out for the night. But why we dream, what function dreams serve, whether they serve any function at all, these questions remain hotly debated. Perhaps dreams are a side effect of the neural housekeeping that occurs during REM sleep, random activations that our meaning-seeking brains weave into narratives. Perhaps dreams serve to rehearse threatening scenarios, a kind of nocturnal fire drill for the mind. Perhaps they consolidate memories, prune unnecessary neural connections, or process emotional experiences. Perhaps all of the above, or none of them. What we do know is that the brain cycles through these stages multiple times each night. A complete sleep cycle, from light sleep through deep sleep and into REM, takes about 90 minutes on average. You will complete 4 to 6 of these cycles in a typical night, and the composition of each cycle changes as the night progresses. The first few cycles contain more slow-wave sleep. The later cycles contain more REM sleep. This is why you often experience your longest and most vivid dreams in the hours just before waking. Now, I have spent a great deal of time talking about what happens in the brain during sleep, but I would be remiss if I did not spend a few minutes talking about what happens when we do not sleep enough, because the consequences of sleep deprivation are, frankly, terrifying. After one night without sleep, your cognitive performance drops significantly. Your reaction time slows. Your attention becomes spotty. Your ability to form new memories is impaired. After 2 nights, you begin to experience microsleeps, brief episodes lasting a few seconds during which you lose consciousness, often without realizing it. These are extremely dangerous if you are driving or operating machinery. After 3 nights, you may begin to experience hallucinations and paranoid thinking. But you do not need to stay awake for days to experience the effects of sleep deprivation. Chronic sleep restriction, consistently getting less sleep than you need, accumulates what researchers call sleep debt. And unlike financial debt, you cannot really pay it off by sleeping extra on the weekends. Studies have shown that people who consistently sleep 6 hours per night, after just 2 weeks, show cognitive impairments equivalent to someone who has been awake for 24 hours straight. And the worst part? They do not realize how impaired they are. They rate their own alertness and performance as only slightly decreased, while objective measures show dramatic deficits. The damage is real, but the warning lights have burned out. Sleep deprivation affects not just cognition, but also physical health. Chronic sleep restriction is associated with increased risk of obesity, diabetes, cardiovascular disease, and weakened immune function. It disrupts the hormones that regulate appetite, making you hungrier, particularly for high-calorie foods, while simultaneously reducing your ability to metabolize those calories efficiently. It impairs your immune system's ability to fight off infections. It may accelerate aging at the cellular level. And then there is the question of neurodegenerative disease. Remember the glymphatic system, the brain's waste clearance mechanism that is most active during deep sleep? When you do not sleep enough, this system does not have time to do its job. Waste products accumulate. Beta amyloid builds up. And some researchers believe this may be one pathway by which chronic sleep deprivation increases the risk of Alzheimer's disease. The relationship is probably bidirectional. Alzheimer's disease disrupts sleep, and disrupted sleep worsens Alzheimer's disease, creating a vicious cycle that may explain why sleep problems often precede the cognitive symptoms of dementia by many years. I tell you all this not to frighten you, though a healthy respect for sleep is appropriate, but to emphasize just how essential sleep is. We live in a culture that often celebrates sleep deprivation as evidence of dedication and productivity. We admire people who claim to need only 4 hours of sleep, who work through the night, who treat sleep as an inconvenience to be minimized. This is deeply misguided. The science is clear. Sleep is not optional. It is not a luxury. It is a biological necessity, as essential as food or water, and the consequences of skimping on it are real and serious. So, to summarize our journey tonight, your brain falls asleep through the coordinated action of 2 systems: your circadian rhythm, which tells your brain what time it is, and your homeostatic sleep drive, which tells your brain how long you have been awake. As night falls and adenosine accumulates, the balance tips from wake-promoting systems to sleep-promoting systems. The VLPO activates, releasing GABA and galanin to silence the arousal centers. Your body temperature drops, your brain waves slow. You drift through the hypnagogic state, past the K-complexes and sleep spindles of stage N2, down into the synchronized slow waves of deep sleep. And there, in the deepest darkness of stage N3, your brain takes out the trash, pumping cerebrospinal fluid through expanded channels to wash away the day's accumulation of metabolic waste. Then you rise again through lighter sleep until you reach the strange activated state of REM sleep, where your body is paralyzed but your mind is alive with dreams. This cycle repeats 4 to 6 times per night, each cycle lasting about 90 minutes, each one slightly different from the last. And when you wake, if all has gone well, you have not merely rested, but have undergone a nightly maintenance routine more sophisticated than anything human engineers have ever devised. Your memories have been consolidated, your emotions have been processed, your brain has been cleaned. You are ready for another day of consciousness, another day of being awake. The philosopher Heraclitus said that we cannot step in the same river twice. I would argue that we cannot wake up as the same person we were when we fell asleep. The sleeping brain changes us, refreshes us, prepares us for the world. Sleep is not a small death, as some poets have suggested. It is a small rebirth, occurring every night, taken for granted until it is lost. So tonight, when you close your eyes and feel that familiar heaviness settling over you, take a moment to appreciate what is happening. The adenosine has accumulated, the VLPO is activating, The flip-flop circuit is about to flip. Your body is cooling, your brain is slowing, and you are about to embark on one of the most remarkable journeys a brain can take. Good night, everyone. Sleep well. Your neurons are counting on it. And in case you've enjoyed our evening talk so much and still might be awake, here is another educational video for you.